I will talk about the core metabolism of microbes, which transforms food into the free energy that drives cellular processes. To analyze how evolution shapes metabolic traits, I considered two aspects of reproductive success. First, how fast do cells grow? And second, how efficiently do cells transform food into reproductive yield? We can often measure those growth rate and biomass yield components of fitness. However, those broad fitness components leave out the fundamental biochemistry of cellular metabolism. What I find particularly exciting are recent breakthroughs in technology. We can, for the first time, measure the small variations in biochemical traits by which natural selection tunes cellular energetics and thermodynamics. My topic today is this possibility of linking natural selection to cellular biochemistry and metabolism. This is all so new that I'm not going to reach any strong conclusions today. Instead, I will emphasize how classic Darwinian principles will, in the near future, allow us to connect natural history to cellular metabolism. To introduce the core ideas that we need for this discussion, I'm going to start with a simple example. Suppose we begin with a vat that contains some sugar, and into that vat we throw a clone of yeast, and the yeast are all genetically identical and from a single clone. And the yeast do what they're very good at, which is suck up the sugar and reproduce, and transform the sugar into biomass. And under this scenario, where we have isolated clones growing, natural selection favors those genotypes that are most efficient at transforming sugar into reproductive yield, because those clones that are most efficient produce the greatest number of progeny and have the highest fitness. Now let's run the same experiment, but with a small variation. We've got a vat with some sugar, and let's throw two different genotypes of yeast into the vat and let them compete for that sugar. One of the genotypes sucks up the sugar faster and grows faster. And so by the time all of the sugar is gone, this faster growing genotype has outcompeted the slower growing genotype. But to suck up sugar faster and grow faster, this particular genotype must expend free energy to bring the sugar across the cell membrane at a faster rate and to drive metabolic reactions at a faster rate. And free energy expended in driving those processes to increase growth rate is not available for reproduction. So necessarily, the faster growing genotype is less efficient at transforming sugar into reproduction. And so fundamentally, there's a trade-off between growth rate and reproductive yield. So overall, when we have clones growing in isolation, selection tends to favor the maximization of reproductive yield, whereas when we have clones growing in competition, different genotypes growing in competition, selection tends to favor high growth rate at the expense of reduced yield. And this trade-off between growth rate and yield is fundamental and is widely discussed in microbiology. And let's start with a picture just to summarize this key trade-off. So the idea is that to grow faster, increase rate, requires the expenditure of free energy to drive rate processes, and that energy is not available for reproduction, and so reduces the reproductive yield efficiency, where yield is the amount of reproduction per gram of sugar taken up. And so we can see there's a broad trade-off between growth rate and yield that could potentially tune metabolic biochemistry. And there's been a lot of discussion of this in the literature, and I'll return to this point later. But conceptually, Pfeiffer et al., Pfeiffer and his colleagues, about 20 years ago emphasized the point that I just made previously about the genetic structure of populations. When genetic relatedness increases, or the genetic correlation between genotypes within a patch increases, this tends to favor high yield, because with high relatedness, there's relatively little competition between different genotypes. And so selection favors the maximization of reproductive efficiency, which improves the success of all members of the group. By contrast, when relatedness declines and the genetic mixing increases, reducing correlation between genotypes, that favors competition between genotypes, which favors high growth rate at the expense of reduced yield. So we see fundamentally that the genetic structure of populations in terms of kin selection or group selection can be a very powerful force shaping the metabolism and biochemistry of microbial cells. As I said, this has been widely discussed in the literature and is quite well understood, and there's some empirical evidence to support this idea. But although this genetic structure argument is widely known, there are many other aspects of natural history which could also influence cellular biochemistry and metabolism that get much less attention. For example, basic principles of demography could also be very important. As shown in this example, same situation, we imagine an isolated patch with some sugar and a clone that grows up and transforms the sugar into reproduction. Here we have two different genotypes, each growing in isolation, but with different growth rate properties. A fast-growing genotype would suck up sugar faster, but would come to a lower final biomass size for its population because it's using more of its energy to grow fast and therefore has less yield per gram of sugar taken up and therefore would produce less biomass for the same amount of sugar. This clone, by contrast, grows up more slowly but goes to a higher final biomass size because it's more efficient at transforming sugar into biomass. Now here we have the different times that are happening for these growth rates, and we imagine that patches occur in nature, and patches can be disturbed by a variety of natural processes. And so we can imagine a patch lifespan. And so here we can imagine this is a demographic property for how long do resource patches last. If resource patches tend to get disturbed after 20 time units, that will tend to favor high growth rate rather than high yield, because faster growing clones will grow as fast as possible before the disruption at this 20 time unit point. In other words, if the cells don't use up all the available sugar before disturbance occurs, they might as well grow as fast as possible because there's no limitation on sugar and no benefit to being efficient at transforming sugar into reproductive yield. By contrast, if patch lifespan is quite long, that tends to favor high yield over high rate because efficiency is favored and leads to the production of a greater number of progeny. So we can see that demography can possibly be a very powerful force tuning cellular metabolism, and this gets relatively little attention in the literature. Now what happens if we take these two genotypes and we put them together and let them compete directly within the same resource patch? In direct competition, high rate beats high yield, because this genotype will grow up relatively quickly, and by the time the slower growing genotype is starting to increase its biomass, all of the sugar is gone. And so the high rate genotype beats the high yield genotype in direct competition. So we can put together the demography and genetic structure arguments, and I did some calculations several years ago to see what sort of synergism there would be between demography and genetic structure. And so this shows you by theoretical calculation the optimal trade-off between rate and yield, where the higher this number, the higher the growth rate, and the lower the yield. That would be a more competitive strategy. Genotypes growing fast to outcompete their neighbors. A low value here is a high yield and slow growth rate, and that would be a more cooperative strategy because high yield tends to be associated with an increase in the reproductive success of all members of a group in a cooperative sense. Now we can see here are the kin selection coefficients or genetic correlations within a patch which describe the genetic structure of the population. One represents pure clonality, where the correlation between genotypes within patches is perfect, and this is more mixing, which reduces the genetic correlation. And we see that the more mixing there is, the more high growth rate is favored at the expense of reduced yield, whereas clones tend to have high yield and low growth rate. Now here, along this axis, is the expected survival time of patches. This is the demographic property of populations. When patches tend to get disturbed relatively quickly relative to the resource flow, so typically disturbed before all the resources are used up, then it pays to grow fast, more or less independently of the particular genetic structure, because the patches will be disturbed before most of the resources are used up, and so high yield and high efficiency provides relatively little benefit. Whereas if patches tend to last a long time, resources often become depleted, and therefore high efficiency is favored. And so we can see a very powerful synergism between demography and genetic structure of populations. And this is generically true for any situation where there's conflict and cooperation potentially, and also a very powerful prediction potentially for how natural history shapes metabolic biochemistry. Now, this is a very pure theoretical argument, and we could continue on with a variety of other principles of natural selection and make several theoretical arguments about how natural selection would shape and tune cellular biochemistry and metabolism. But today, instead of developing the theory extensively, what I want to do is turn to some recent studies on cellular biochemistry and metabolism and see what opportunities we may have to connect these general abstract principles of natural selection to the actual way in which natural selection shapes biochemistry. So let's turn to some data and particular observations. And one of the common observations in microbial metabolism is this particular puzzle known as overflow metabolism. And this occurs quite widely across microbes under certain circumstances. So here's the idea. This is core metabolism. Cells take up sugar, break up that sugar through glycolysis and produce pyruvate. Pyruvate is then transformed into molecules that get fed into the TCA or citric acid cycle. And the TCA cycle produces the molecules disequilibria that drive electron transport and oxidative phosphorylation. And so this core metabolism is basically the way in which all of life transforms food into the free energy that drives cellular processes. There's a lot of variation, but this core metabolism is quite consistent as a sort of core structure throughout the history of life. Now, interestingly, when cells start to grow quickly, they often dump fermentation products such as acetate or ethanol right from glycolysis. And this is a puzzle because only about 10% of the ATP that can be produced from taking up sugar is made in glycolysis, and about 90% of the ATP is produced down here. So when food is dumped at this point or excreted at this point, then most of the free energy in the food, the usable free energy in the food, is being excreted rather than being used. And this is a puzzle because why would cells excrete most of the usable free energy in their food rather than using that free energy to drive metabolic processes? But we see this quite widely when cells are growing quickly. They start to excrete these overflow products. So there's a very big literature discussing the nature of overflow metabolism. There was a very nice study published just a few years ago in Nature by Bassan et al. I'll briefly tell you about this. So overflow metabolism happens in many different, in yeast and bacteria. It also happens in human cancer cells. And so it's quite a widespread phenomenon, but they studied E. coli in the laboratory. And these different symbols represent different strains of E. coli that were taken into the laboratory and grown under different conditions. And all of these different strains fall along this simple line here that relates growth rate to a degree of acetate overflow, the amount of acetate that's excreted from the cells during metabolic process. And we can see that when cells grow relatively slowly, they do not excrete any acetate. But as the growth rate increases, they increasingly excrete these overflow product as acetate, so from glycolysis. And so those are the basic observations. And excreting acetate is associated with declining yield because for a given amount of sugar intake, the more acetate that's overflowed, the more of the usable free energy in the food is being excreted, and that reduces the yield as measured as the amount of reproduction per gram of sugar taken up. So we see here a mechanistic coupling between growth rate and declining yield associated with acetate overflow. And so that's the puzzle. And Bassan et al. Did a number of studies to try to figure out what might explain this great puzzle in microbial cellular metabolism. And their conclusion is proteome limitation or protein limitation. The idea is that cells can only contain a certain amount of protein. There's a limit on how much protein can a cell can contain, because there's a limit on the volume of a cell. And this protein limitation then works as follows. Proteins are limiting. There's a certain amount that can be allocated. Some of those proteins can be allocated as catalysts that drive the TCA cycle, which drives electron transport. But proteins that are used for this component of core metabolism are not available for anabolic processes for growth. And so the proteins then form a constraint that causes a trade-off between allocating more proteins for efficient metabolism or allocating more proteins to increase growth rate. And so the fundamental growth rate versus yield trade-off comes from a fundamental biophysical limit on the amount of proteins contained in cells. And they did a number of very well-designed manipulative experiments in the laboratory to show that a quantitative model based on this biophysical limit of proteins predicted very well what happened when they perturbed this protein compartment. For example, they could reduce the available amount of protein in the cell and make a prediction about how cells would reallocate proteins, and they got a very close match to such predictions. And so they essentially claimed that they've solved the general puzzle of overflow metabolism. It really comes down to an essential biophysical limit, and that this would be true for bacteria, for yeast, where overflow metabolism is known as the Crabtree effect, and for human cancer cells, where overflow metabolism is known as the Warburg effect. And I think this is a very compelling argument, but there's an aspect of it which is limiting or not quite complete, which is that if we try to think about how evolution designs organismal traits, biophysical limits are very important. Organisms cannot violate biophysical limits, and those constraints set very important trade-offs on the nature of organismal design. On the other hand, we know that natural selection can widely tune the traits of organisms, and so I'm always suspicious of an argument that only focuses on biophysical limits, because we know when we select on traits, we get a very wide response. And so the best way to think about traits is that there's always a duality. There are always biophysical limits, but natural selection almost always can tune metabolic traits or organismal traits within those limits. And so we need to look at both sides of this duality, the biophysical limit side and the natural selection side. And so now I turn to a study that looked at how natural selection can tune biochemistry. This is a study done in Lactococcus, which is an anaerobic bacterium. And this will go back to the argument about rate versus yield trade-offs in relation to the genetic structure of populations. So this is an anaerobic species, so it takes up glucose and goes through glycolysis to make pyruvate, and then the pyruvate is excreted as a fermentation product after a transformation. This particular pathway transforms pyruvate into lactate and excretes the lactate, and that's associated with an overall yield of 2 ATP per glucose molecule. And so this is a relatively low yield pathway compared to this pathway, but it's associated with higher growth rate. So we see biochemically going this way is high growth rate, low yield, or the pyruvate can be transformed into these fermentation products, acetate or formate or ethanol. That makes an extra ATP, which is a higher yield per gram of sugar taken up, but it is associated with a slower growth rate. And indeed, the cells that go through this pathway have slower growth rate, but greater reproductive efficiency. So we can see that single cells are capable of alternatives. And essentially, this is like overflow metabolism. Here, the cells are excreting more usable free energy, but associated with that, they're gaining an increase in growth rate. Now, to look at how natural selection would tune the different pathways and the different strategies of rate versus yield, they made a knockout of lactose dehydrogenase, which knocked out this pathway. And so that strain can only go through the high yield, low rate pathway. An alternative strain almost always goes through the lactate pathway, which is high growth rate, low yield. So they could compete these two strains under different conditions and look at the conditions that might favor one strategy or the other. How does natural selection tune biochemistry? And as I said, they were focused on this genetic structure argument, and they came up with a clever experimental design to vary genetic structure. In one experiment, they used an oil-based habitat that had water droplets within the oil. So it's an oil emulsion with water droplets. And when they put the bacteria into this particular habitat, a small number of bacteria colonize each water droplet, which also contains some sugar. And so basically each isolated patch is a clone with no genetic variation. Under that circumstance, they predicted that the high-yield, low-growth rate pathway would tend to be favored. And then alternatively, they grew the two different genotypes in direct competition, and that they believed would favor the high growth rate, low yield. And that's exactly what they saw. When they grew the alternative strains in this emulsion process, this is the percentage of slow-growing strains. That's slow growth, high yield. This is the initial percentage of the mixture, about 25%. And we can see under this situation that the slower growing strain goes to fixation over a course of a few generations, showing the very powerful selective pressure that shapes biochemistry favoring the high yield, low growth rate strain. When the strains are grown in direct competition, the high yield strain loses and the fast growth rate strain wins very quickly. So the suspension is a mixture of genotypes. And so this shows experimentally how we can look at the role of genetic structure in shaping biochemistry. So that's a very powerful argument, but here we're just looking at fixed genotypes with particular biochemistries. One question that arises is, what happens if we select on growth rate? How does that affect the tuning of cellular metabolism? And so an experiment was done a couple of years ago, and so let's return back to the basic observations on E. coli. These are strains that were obtained, different E. coli strains obtained and grown in the lab under different conditions, generally with different amounts of sugar availability. And we can see that all of these strains fell along this rate versus yield trade-off associated with acetate overflow. Now in this new experiment that was published just less than 2 years ago, here's the same graph, growth rate versus acetate overflow. These are the data I just showed you. All the different strains that have been obtained fall along the line. But to start their experiment, they started with a strain that's down here that has a slightly different growth rate and acetate overflow property. And their experimental protocol selected for high growth rate. So they wanted to see what happened when they imposed selective pressure on strains for high growth rate. And so these are the outcomes. They had a number of independent replicates of the experiment. So each of these represents the outcome after many generations of evolution in an independent replicate. All of the strains greatly increased their growth rate, but you can see that they fluctuated quite widely in the acetate overflow and the amount of yield, because higher acetate overflow is associated with lower yield. And so we can see that yield varied quite widely, even though growth rate tended to be attracted to something that seems to be a maximum here. And they suggested that that seems to violate the basic growth rate versus yield trade-off. These are the prior observations from the literature, where we can see that the growth rate and yield fall pretty well along the line, the trade-off line. But in their experiment, they started here, and these are the outcomes. And you can see the outcomes had high growth rate, but the yields were fluctuating in a more or less neutral manner. And they suggested that was a bit of a puzzle, because this growth rate versus yield trade-off was thought to be a really fundamental property of cellular metabolism, and previous studies fell along that trade-off. But here they seem to have broken the trade-off, and they suggested that was a bit of a puzzle and didn't give a very clear explanation for that. But I think I know what's going on in this case. In their experimental protocol, they transferred the E. coli growing in a particular medium to the next fresh batch of medium before all the sugar was used up in the initial medium. And that means that the cells are never facing a limitation on sugar and food. And because they're not resource limited, there's really no selective pressure for efficient yield. And so yield's effectively a neutral trait. So they selected on growth rate and got high growth rate, but their experimental protocol left yield to be neutral. Basically, the demography that they used for the aging of populations in relation to the flow of resources did not select at all on yield. Now, presumably, they could change that and select also on yield by altering the demography. And as we know in natural history and natural populations, demographic aspects vary quite widely. And so what I'm arguing is that variations in demography in natural populations are likely to be very important in tuning fundamental biochemistry of metabolism. And there's some evidence for that. I don't have time to discuss it from laboratory experiments. So demography is likely to be very important. Now, I've been emphasizing biochemistry all along during the talk, but in fact, I've mentioned relatively little about biochemistry. And so let's turn to another study that was published just last year from a laboratory from UCL. And this is really a very nice study that's starting to get down into a lower level biochemistry and looking at how natural selection might be involved in biochemical variation. And so in this study, this was done in yeast, and they collected a large number of isolates from natural populations and also some laboratory isolates. I believe there were about 160 isolates or different strains that were evaluated. So in this yeast species, and they discovered among the different isolates a polymorphism in a single important allele that's involved in glycolysis, and that's the pyruvate kinase gene that transforms this particular molecule into pyruvate, PYK1. And they found a high activity allele and a low activity allele. And here I'm showing the results for the high activity allele from their study. So these are steps in glycolysis. This is fermentation overflow, so you see acetate or ethanol. And comparing the high activity allele relative to the low activity allele, green represents a higher concentration of a particular intermediate or a higher concentration of a particular enzyme in case of these symbols here. And red represents decreased expression or decreased concentration. And so you can see that this polymorphism had a wide effect on tuning biochemistry in terms of the concentrations of different intermediates and the concentrations and expression levels of different enzymes. Now, I was just showing you a subset. I was showing you the glycolytic subset up here with overflow. And the high activity allele actually had greater overflow than did the low activity allele. And this is the rest of the core metabolism, the citric acid cycle and electron transport and oxidative phosphorylation. And we can see then that they measured and showed quite broadly the variations associated with this polymorphism. In particular, you can see the great downregulation in electron transport and oxidative phosphorylation associated with the high activity pyruvate kinase allele. And so the point is that they measured tremendous variations in the tuning of the cellular biochemistry in relation to a natural polymorphism. Now, they also looked at some of the growth rate characteristics of this polymorphism. What they found is that the high activity pyruvate kinase allele had a higher growth rate, but it had a lower yield and was actually associated with greater overflow, which is associated with lower yield. And so actually this allele was tuning the growth rate versus yield trade-off and the degree of overflow metabolism. And it was polymorphic in natural populations, suggesting that certain natural history conditions would favor one aspect or tuning of biochemistry, and other habitats would favor a different tuning of fundamental biochemistry. Now, interestingly, there was another fitness component that varied, and that was tolerance to reactive oxygen species or tolerance to oxidative stress. The high activity allele with the faster growth rate was less tolerant to oxidative stress. Oxidative stress is a very important selective component in natural population, natural habitats, and can vary quite widely. So this can be a very important fitness component. And so this emphasizes that looking at just rate and yield is often quite misleading because these rate and yield components can be coupled with other very important fitness components, in this case oxidative stress tolerance. And so the key point here is that we're seeing how we can begin to measure fundamental polymorphisms that vary in natural populations by habitat, and we presume that natural selection is working on these polymorphisms to tune cellular biochemistry. So we're beginning to get a notion of the potential to connect natural history to the tuning of cellular biochemistry. And so this is really an important study showing what I think we can look forward to in the future. Now, one other aspect of their study I found very interesting, which is they looked at the NADH, NAD+ disequilibrium in the cell. And I don't have time to go into this, but this disequilibrium is really one of the key measures of the thermodynamic state of a cell, which is really the central notion of metabolic biochemistry in terms of free energy and thermodynamics. The high activity allele had a much greater disequilibrium than did the low activity allele. And this is something that's been seen in prior studies, that fast-growing cells that are overflowing their metabolism tend to have a strong NADH, NAD plus disequilibrium. And so this suggests the potential to connect the observations on polymorphism to the thermodynamic state of cells. And so now we're really getting down to the fundamentals of free energy flow within cells and how natural selection is tuning that. And so in the last part of the talk, the very last point, I want to turn to this notion of free energy, because this is really the essential notion of cellular biochemistry, is getting down to the free energy and entropy aspects of metabolism and biochemistry. And so here's a simple argument about the free energy. This is the free energy delta G. And here we're showing two coupled reactions. The top reaction has all reactants and no products. And so there's going to be a strong thermodynamic driving force pushing the reaction to go forward. Or you can say an entropic driving force. So the free energy change is going to be largely negative. The greater the negative free energy change, the stronger the driving force to push a reaction faster. Reactions only go forward when they have a negative free energy change. Here's a second reaction that's coupled, and it's more or less in equilibrium, and so driving this reaction forward actually causes a positive change in free energy, which would not happen spontaneously. But if these two reactions are coupled biochemically, then this driver reaction can push this other reaction to go forward and create a disequilibrium. And this is actually the fundamental way in which metabolism works and how basically all of life works. For example, if we think about a reaction isocitrate oxalosuccinate as a driver reaction, that can drive the NAD plus NADH disequilibrium. And this disequilibrium then is used to drive other cellular processes. And here the net free energy change remains negative because this is very negative and this is slightly positive. And when you have coupled reactions, you have to add the free energies. And so this driver reaction can drive this disequilibrium. And that's the fundamental notion of metabolism. Indeed, you can think of metabolism such as the steps in glycolysis as each reaction is a drop in free energy, which must be so, otherwise the reaction would not proceed, and that the degree of drop in free energy depends on the yield, the degree to which a particular reaction is driving a disequilibrium in NADH or in ATP. The greater the yield, the less the total free energy change, because these are positive contributions to free energy, and the driver reaction's a negative contribution. And so we see here the fundamental thermodynamic notion of rate versus yield. The bigger the free energy change here, the greater the driving force pushing the reaction faster, and the faster the reaction goes. But the greater the free energy change, the less the capture of the total free energy as ATP or NADH, which is the yield. And so we see fundamentally the trade-off between metabolic reaction rate and the yield of metabolic reactions. And so the real goal in metabolic biochemistry for cells is to tune this process of drops in free energy to get a high level of yield, but to maintain a fast rate of metabolism, given the fundamental thermodynamic trade-off. Now here, if we imagine the driver reactions in equilibrium, and this reaction is already in disequilibrium, this reaction will not go forward. In fact, there's a pressure to go in the reverse. This is, you might call this thermodynamic inhibition. This would actually drive the metabolic reaction in reverse and would halt the metabolic cascade. And so overflow metabolism is actually, when this sort of situation happens, cells tend to overflow reaction products in order to relieve thermodynamic inhibition. Now, one of the great technical breakthroughs in the last couple of years is the ability to measure the delta G, the free energy change for reactions in vivo. Traditionally, free energy changes were very difficult to measure. They were measured in vitro with a lot of assumptions about how things were working. But actually, these were probably rather poor estimates of actual in vivo thermodynamics. And without measurements of in vivo thermodynamics, we actually did not have a very good sense of how cells were tuned biochemically. But the breakthrough is that it's now been possible to label reactants and products and measure the net forward flux and reverse flux in vivo. And from the ratio of the forward to reverse fluxes, it's been possible to estimate the free energy changes. And so then, people have been able to estimate the actual free energy changes associated with the various reactions and get a much better sense of the biochemistry and how biochemistry actually works metabolically. And this leads us to the possibility that we could look at how genetic variants could tune the free energy changes for particular reactions and the yield captures, and then we could get a sense of how natural history would alter the basic tuning of metabolic biochemistry. And all of these things potentially now are measurable. This is very new, and so there's relatively little that's been done. Here's one study that looks at 2 different species and measures the thermodynamic changes for 6 steps in glycolysis. In this species, which grows very slowly, the free energy changes are very small. There's a very slow rate of growth. But associated with that, this particular species makes an extra ATP through these glycolytic steps, compared to this other species. Both species are of Clostridium genus. This other Clostridium has much greater free energy changes, has a higher rate of glycolytic throughput, but has a lower efficiency and makes 1 less ATP through this metabolic cascade. And so we can see the reaction rate versus efficiency yield capture being played out in a measurable way in individual cells. Now, if we can imagine genetic variation within a species that's tuning these particular reactions and affecting the metabolic throughput and also the yield. And we could connect those variations with different selective pressures that happen in natural history. And so that's what I imagine is going to happen in the next 10 or 20 years, is this connection between natural history and metabolic biochemistry. And so I've just about completed a draft of a new book that I call Microbial Life History. The first part of the book goes over the fundamental forces of biological design, the basic theory by which we understand how natural selection and natural history tune traits and affect the design of traits. And then we look at the potential for connecting those general principles to the actual design of metabolic biochemistry by looking at the current state of the art in the observations of microbial metabolic biochemistry. And as I've emphasized, this is all still quite new, and so the book is really about the potential for making this connection and the kinds of conceptual breakthroughs that we need to test the proper hypotheses to connect natural history and natural selection to the design of metabolic biochemistry. Thank you.